Digestion. In mouth, lingual lipase is secreted by Ebner's gland and on the dorsal surface of the tongue, but it has no action in the mouth. In the stomach, lingual lipase is active in stomach because the action starts in acidic pH. It digests about 30% of the dietary triglyceride. Gastric lipase is secreted in stomach but has little importance except in pancreatic insufficiency. In small intestine, most of the fat digestion begins in the duodenum, pancreatic lipase being one of the most important enzyme involved. The digestion of the fat includes three steps, that is emulsification of the fat by bile salts, then hydrolysis of fat by pancreatic and enteric lipolytic enzymes, then acceleration of fat digestion by micelle formation. Emulsification of fat by bile salts. The first step in fat digestion is physically to break the fat globules into very small sizes so that the water soluble digestive enzymes can act on the globule surfaces. This process is called emulsification of the fat. Emulsification of the fat mostly occurs in the duodenum under the influence of bile. Bile contains large quantity of bile salts as well as phospholipid lecithin. Both of these, but especially the lecithin, are extremely important for emulsification of fat. The polar parts of bile salts and lecithin molecules are highly soluble in water, whereas lipophilic non-polar portions of their molecules are highly soluble in fat. So this is the emulsification, where the natural fat, it will be surrounded by the bile salts, and then bile salts is going to convert the larger fat globules into the smaller fat globules, so that surface area for the action of enzyme is going to increase, and this is called as emulsification of the fat. Therefore, fat soluble portions of these liver secretions dissolve the surface layer of the fat globules with the polar portions projecting. The polar projections in turn are soluble in surrounding watery fluids, which greatly decreases the surface tension of fat and makes it soluble as well. With the lowered surface tension of fats, segmentation movements of small intestine break up large fat globules into fine droplets. The surface area available for the action of lipase is increased many thousand times by emulsification of fat. Then hydrolysis of the fat by pancreatic and enteric lipolytic enzymes. The pancreatic lipase hydrolyzes the triglycerides, that is, triglycerides gets converted into free fatty acids plus 2 monoglycerides. Then colipase, it binds with the pancreatic lipase and increases its lipolytic activity, whereas bile salt activated lipase catalyzes the hydrolysis of cholesterol esters of fat soluble vitamins, phospholipids, and triglycerides. Then cholesterol ester hydrolase hydrolyzes the cholesterol ester to cholesterol and fatty acids, whereas phospholipase A2 hydrolyzes the phospholipids. Enteric lipase, the enterocytes of the small intestine contain small amount of lipase known as enteric lipase, but this is usually not needed. Acceleration of fat digestion by micelle formation. The hydrolysis of triglycerides is a highly reversible process. Therefore, accumulation of monoglycerides and free fatty acids in the vicinity of digesting fats quickly blocks further digestion. But the bile salts play additional important role in removing monoglycerides and free fatty acids from the vicinity of the digesting fat globules almost as rapidly as these end products of digestion are formed. Bile salts, when high concentration, the concentration is called as critical micellar concentration, have the tendency to form micelles. They are small spherical cylindrical globules containing 3 to 6 nanometers in diameter and composed of 20 to 30 molecules of bile salts in such a way that the lipid soluble non-polar ends are in the central fat globule and water soluble polar ends fan out to form outer covering micelle. covering of the micelle. The monoglycerides and fatty acids are released from the digestion of the fat, are quickly incorporated into the central fatty portion of the micelle, forming which is known as a mixed micelles. In this way, bile salts accelerate the fat digestion by allowing the lipolytic action to continue. So here, this is the mechanism how the bile salt is going to, surround the lipids and by making them water soluble and to enzymes to act on them, and hence they are going to facilitate the digestion of the fats. The bile salts micelles also act as a transport medium to carry the monoglycerides and free fatty acids, both of which would otherwise be relatively insoluble to brush borders of the intestinal epithelial cells. There, the monoglycerides and free fatty acids are absorbed into the blood, but the bile salts themselves are released back into the chyme to be used again and again. Absorption of fat. Normally, through lipids are thought to enter the enterocyte by passive diffusion. Evidences are there regarding the involvement of carriers. In enterocytes, the fate of free fatty acids is as follows. Fatty acids less than 10 to 12 carbon atoms are water soluble, and they pass through the enterocyte unmodified and are actively transported into the portal blood. They circulate as a free fatty acids, that is unesterified fatty acids. Fatty acids with more than 10 to 12 carbon atoms are reesterified into triglycerides. The absorbed cholesterol is also esterified. Triglycerides and cholesterol esters are then coated with a layer of proteins, cholesterol, phospholipids to form chylomicrons. They leave the cells and enter lymphatics, that is in the mucosal cells. Triglycerides are formed from two monoglycerides in smooth endoplasmic reticulum. Some are formed from glycerophosphate, that is the product of glucose catabolism. Glycerophospholipids formed from the glycerophosphates and also participate in the chylomicron formation. The finished chylomicrons are excluded from exocytosis from the basal or lateral aspects of the cell. The free fatty acids are diffused into the portal blood, while the lipids from the chylomicrons are diffused into the lacteal and then through the thoracic duct into the circulation. Absorption of long chain fatty acids is the greatest in the upper part of the small intestine, but appreciable amounts are also absorbed in the ileum. Note that short chain fatty acids and medium chain fatty acids are directly absorbed from the intestinal lumen into the portal vein and is taken to the liver. Short chain fatty acids have 2 to 5 carbon atoms. They are formed by the action of colonic bacteria on the complex carbohydrates, resistant starches, and other components of the dietary fibers. Thus, short chain fatty acid is produced in the colon and reabsorbed from it. So this is showing digestion and absorption of the fat, but triglycerides converted into monoglycerides and free fatty acids, they are going to form the micelles and the monoglycerides. Ultimately, the monoglycerides, free fatty acids, they are going to be converted into chylomicrons in the enterocytes, and they are released into the circulation. They enter into the blood through the lacteal lymph vessel system.